You know what, we'll just do without the title. All right, thanks to the organizers for inviting me to speak. Today, most of the work I'll be talking about was done by my PhD student, Nelson, who wasn't able to make it today. And so the basic premise of our work here is that we're really interested in understanding how do cells respond to different kinds of stimuli. And so the basic premise is that an ideal experiment that you'd want to do is to be able to take a cell, somehow quantify its transcriptome, and then simultaneously perturb that cell with some stimulus, and then measure the transcriptome again, and somehow basically be able to measure, say, for example, differential expression on the exact same cell, on the exact same cell under two different conditions, in order to understand what the stimulus is doing. But of course, this isn't really possible because for most protocols, we have to lyse the cell and get the RNA out, and so you can't do the same experiment on the exact same cell. And so obviously the next best thing you can do is basically do something like single cell sequencing on a bunch of cells before stimulus, a different set of cells after stimulus, and then try to compare them in order to figure out what the stimulus is doing to each cell. And so if you do this and you draw, for example, your standard Tst visualization, what you'll generally find is that cells separate by their condition. And so this obviously prevents us from directly comparing cells under one condition to its corresponding cell state in the other condition in order to figure out what happened. And so solving this problem of figuring out which cells in one condition map to another is a problem generally called alignment or data integration now. And so the basic idea is that, for example, here what I've shown you is what happens if you take cells from two different conditions and you do something really simple like just subtract the mean effect of the condition from every gene. And what you'll see on the right here is that on the right here are three different plots corresponding to three different cell types in this data set. And what you'll see is that the red cells basically overlap after kind of correcting for the mean effect of the stimulus. But for your green and purple cells, they don't really line up after correction for stimulus. And so this alignment problem is hard, basically when different cells respond differently to the stimulus. And so there's been a number of methods developed to deal with this alignment problem. The way that we think about it is through an unsupervised transfer learning approach. And so the basic idea here is that we're using something like a deep autoencoder to generate an embedding of each cell into some low-dimensional space. The main difference between what an autoencoder generally does and what we're doing is that with a standard autoencoder, you might be finding some embedding that minimizes your reconstruction loss if you're trying to predict your input using your output. Whereas what we do actually instead is that we generate an embedding which minimizes the difference between two similarity matrices. So you could imagine that in your original gene expression data, if you're looking at, say, here in our toy Tst diagram, I've colored cells based on the condition that we sequence them in. And so you can, taking your single cell data and looking at the original gene expression measurements, what you can do is you can build a pairwise cell-cell similarity matrix where basically for each cell you look at other cells in the same condition, and you basically, you get higher similarity if your gene expression profile is more similar. And you can also build a similar a matrix using the embedding space. And so what we do is we use this autoencoder, we take cells, we project them down to a low-dimensional manifold, and then we compute a similarity matrix in a slightly different way. So what we do is that, for example, if we take a cell from the red population, what we can do is we can perform something like a random walk, where we say, for example, started a cell, and we force ourselves to make a walk to a cell from the other data set, where we choose that cell based on how similar you are in the embedding space. And then once we're sitting in this sort of blue data set space, we perform another random walk back to the red cells by basically walking to a cell based on how similar that cell is in the embedding space. And so this kind of round trip probability in this random walk can be used to generate another sort of similarity matrix. And so basically these two similarity matrices basically look different because obviously the embedding space is smaller than the gene expression space, and we try to minimize the difference between those two similarity matrices. And so in that way, we're using this autoencoder also in an unsupervised way, but we're kind of minimizing the distances between these two different data sets. And so we started out, so this study was really motivated by looking at basically cells from this one study where they sequenced three different types of cells, long-term, short-term HSCs, as well as multipotent progenitors. And they did it under two different conditions, basically control mice versus mice that were basically stimulated with LPS. And so we wanted to see whether our autoencoder could basically bring these two data sets together. And so here on the right is basically the T-SNE diagram you get if you just take like an autoencoder or a T-SNE and you just project the cells down to some low-dimensional space and visualize them. And so here on the right is basically the T-SNE visualization you would get. And I'm sorry for spamming T-SNE so often, but you're going to see a lot of these. And so this is the visualization you would get if you use our autoencoder to project these cells into the same space. And what you generally see is that cells from different conditions basically overlap very strongly in the low-dimensional embedding. Of course, it's not very meaningful to overlap in the low-dimensional embedding if you're not lining up cells of the same type. And so here on the right, I'm showing you the same projection, but the cells are colored by their cell type that were defined experimentally. And so what you can see generally is that cells of the same type end up in the same space after using our autoencoder. And so here's just a diagram of the similarity matrices of the data in kind of gene expression space and the embedding space. And again, what you can kind of see by construction is that the embedding space that we learn basically generates a similarity matrix of the single cells that looks very similar to what you get when you look at the raw gene expression data, even though we're doing this kind of weird alignment with the autoencoder. And so we're not the first ones to think of solutions to this alignment problem. And so we basically took a number of existing algorithms that have been developed to deal with this alignment problem and benchmarked them against different data sets. And so here, basically these data sets mainly differ. They all contain the same number of cells, so it's always two conditions. And in this case, there's 3 cell types in each condition. But what they differ in is basically the amount of heterogeneous response of each cell type to the stimulus. And so in CellBench, basically there's not a whole lot of differences in the response of each kind of cell line or cell type to the response. Whereas in HSC het on the very far right, the cell types are, there's very little overlap in the differential expression of the same cell types across condition. And so what you can generally see is that SC-align, the name of our autoencoder, basically seems to consistently perform well, whereas the other methods, they do fairly, like some of them like Seurat do fairly well when the problem isn't that difficult. But when you end up with cell types that respond very heterogeneously, that's where we kind of really shine. And so here's just some visualization showing the cells after alignment. And so basically the cells on the far left are the sort of this very heterogeneous case. And you can see that, for example, with methods like Seurat, the same cell types, basically the cell types don't really separate out as well as they do for SC-align. And so we did some experiments to try to figure out, why exactly this autoencoder and random walk-based approach seems to do better than others. And so we did a series of experiments where, for example, we took cells from one condition and we just started reducing the number of cells in one of the types, right? And so we kind of simulated the case where maybe one cell population is really abundant in one condition, but not very abundant in the other condition. And so here on each one of these graphs, each graph represents what happens when you remove cells from one type. And as you move further to the right in this graph, you're removing more and more of the cells in each condition. And so what you can generally see is that SC-align is somewhat robust to basically removing cells from a given condition. Other methods actually don't do so badly for example, the short-term or even the MPP cells, but for the long-term HSCs, for example, there's definitely a degradation in performance as you kind of remove some of those cells from one of the conditions. And so what I'm showing you on the bottom here is a heat map, which is basically the similarity matrix for one condition of the data set when you've removed 75% of the cells or 25% of the cells. And the main point here is that these two similarity matrices look pretty similar, even though one of the data set has half of the cells removed more than the other. And so SC-align is pretty robust. And the reason it's robust is that if you actually, so I mentioned earlier that SC-align does this kind of two-step random walk. And so this is basically a diagram which shows you basically which cells tend to get visited during this random walk. And so the top line in this graph is basically the starting point of a random walk, and the bottom line is basically the cell you walk to. And what you basically see from this visualization is that many cells basically visit a very few number of cells in the other condition. And so it's robust because if you're just randomly selecting cells to remove, your chances of hitting a hub in this graph is basically very small. And so we applied this method to a number of different data sets, one of which was basically data collected on a study of malaria sexual commitment. And so here the idea is that we know that the gene AP2G is a master regulator of sexual commitment, and its expression is a necessary condition for commitment. And so what happened is that basically the CAPSAC lab from Weill Cornell sequenced two versions of a malaria cell line where AP2G is either allowed to be expressed or completely knocked out. And so the idea here is that we want to see which genes kind of also change with AP2G being knocked out to figure out what other genes may be driving commitment. And so basically what happens is that, so here's a plot of the single cell data generated from this AP2G on, where cells are allowed to sexually commit, or AP2G off, where they're prevented from committing. And so what you can see here on the right is that when you align cells from these two different conditions, you generally get a mix of these red and blue cells, which means that cells of the same type tend to be shared across the two conditions. But what you'll see on the very far right is a small population of cells, which I've labeled as gametocytes, which are only present in the blues, which are only basically represented by the blue cells. And so that kind of shows the SCA line is able to kind of maintain condition-specific cell types, whereas if you look at SIRAD or basically any of the other methods that we've tested, you'll see a very clear mixing of the red and blue dots near where the gametocytes sit, indicating that other alignment approaches may not be able to maintain condition-specific cell types. And so one of the main, novel features, or somewhat novel features of SCA line is that we not only care about generating and embedding where the cells from different conditions overlap, but we actually also train a decoder which projects cells from this joint embedding back into the original conditions, and that allows us to kind of do this kind of simulate basically a paired differential expression. And basically what we found is that when you do this paired differential expression across these two conditions, we actually are able to find a rare cell population that is expressing many genes very differently in the off versus on condition. And we did some further validation follow-up to make sure that those genes are actually involved in sexual commitment. So just to summarize, basically SCA line is an unsupervised framework for doing alignment, although I didn't get a chance to talk about it, but it can actually use labels or partial labels or even overlapping labels. It seems to be somewhat robust to the existence of condition-specific cell types, and it allows us to kind of project cells from the joint embedding back to the original gene expression space to do some simulations of differential gene expression. And this work was supported by a Chan Zuckerberg grant as well as an NIH T32. And I'll be happy to take any questions.
Great talk. I just have a quick question. There was a new method from near Youssef Lab, single cell variational inference, where they are not advertising as alignment, but they jointly, when you combine the two data set, they can actually project it in a lower dimensional space such that it aligns well. I was wondering if you compared to that method as well, or because
so you're talking about the one that just came out a week ago, right?
More than a week ago. I think it's been a few months now.
Oh, okay.
Yeah, it's SCBI. Single cell variation.
Yeah, we haven't benchmarked against that one specifically yet.
Okay.
But yeah, that is we are planning to do so.
Okay. Thank you.
Cool talk. I wonder if you've considered using something like SCA line to look at sort of imputation as a side task, for overcoming things like technical dropout. Because it looks like you can do, imagine sort of you have several conditions, you want to do alignment, and in one of the conditions, you're really actually interested in making some sort of statement about gene expression, but let's say it's in drop where you have an excessive amount of dropout. Do you think you could actually use ST-align to do imputation in one condition or possibly the other?
Yeah, it's a great idea. Yeah, we just haven't got around to doing that yet, but it's definitely in the pipelines. But yeah, it is an obvious thing to do, like project back to the gene expression space and see what happened to the zeros.
Yeah, I just want to ask a question that your loss function is a cell-cell similarity matrix, or is it
so it's the KL divergence row-wise between your random walk probabilities in gene expression space or in the embedding space,
so
where the actual random walk probabilities are defined by it's basically just a scaled similarity matrix.
So what's the input? So is it a count, or do you assume it must be a normal distributed?
It's just the raw counts, the gene by cell matrix.
I see. So you're not assuming it's a zero inflate negative binomial, like variational autoencoder?
No. So we're not. So unlike SCVI, which is a proper graphical model, ours is. We're not using like a negative binomial assumption.
So
or
just
anything
fully
like
connect layer for.
Yeah, okay. And we with like log +1 transformation of the counts.
Just last question. So would you be able to tell the alignment, which is a batch effect, which is biological, meaningful?
No. So yeah, right. So what you're saying is when you have cells sequenced under two conditions, there's the differences due to both batch and biological variation. No, we can't, because you actually, to distinguish those kinds of sources of variation, you need an actual model of what batch effect is or what biological variation is, and we don't have such a model in our aligner.
Thank you.